Welcome everybody again to another episode of the ideaXme show, the show that profiles the humans behind the big ideas that have shaped our world, inspire the future, and for future creators, for all those that like great stories. I'm Ira Pastor, your life sciences ambassador along for the journey today. So as many of you may know that watch the show, aside from the time I spend doing these interviews, my day job is in the biotech sort of regenerative medicine arena. And, every now and then I'll have someone approach me that, is either in the industry or wants to get into it and basically ask the question, what resources should I go pick up if I really want to learn what this space is all about? It's interesting to me, but I'm starting from scratch and I want to get my idea wrapped around regeneration and regenerative biology. Whenever anyone asks me that question, I say, look, there's only one resource that you need at the beginning of this journey, and here it is: Principles of Regenerative Biology by Dr. Bruce Carlson. And today I'm truly honored to be joined by the author of this encyclopedic text, a true living legend among the regenerative biology world, Dr. Bruce Carlson, who is Professor Emeritus of Anatomy and Cell Biology from the University of Michigan. Dr. Carlson, after receiving his master's in Ichthyology at Cornell, completed both his medical and doctoral studies at the University of Minnesota, and over the last 40 years was a faculty member of the Department of Anatomy and Cell Biology at the University of Michigan, and he also directed the Institute of Gerontology. His research over the years has involved limb and muscle regeneration, limb embryology, the biology of aging and denervated muscle. He has authored over 200 papers, 13 books on regeneration, embryology, and lake biology, and edited another 15 symposium volume and translations. He's received numerous awards over the years, including the American Association of Advancement of Science Newcomb Cleveland Prize, the Henry Gray Award of the American Association of Anatomists, where he served as president, and membership in the Russian Academy of Natural Sciences. Over the years, Dr. Carlson has conducted research for extended periods in the former Soviet Union, Czechoslovakia, the Netherlands, Finland, and New Zealand. And in addition to Principles of Regenerative Biology, other books that Dr. Carlson is involved in authoring or co-authoring include Stem Cell Anthology from Stem Cell Biology, Tissue Engineering, Cloning, Regenerative Medicine, Biology, Human Embryology and Developmental Biology, the Human Body, Linking Structure and Function, and Beneath the Surface, Natural History of Fisherman's Lake. Dr. Carlson currently serves on the Scientific Advisory Council of the Fetzer Franklin Fund, whose mission is to explore the frontiers of scientific knowledge and advance breakthroughs towards scientific views of reality that are integrated and relational. All that being said, Dr. Bruce Carlson, thank you for taking the time out of your schedule to join us today. It's a real honor.
Oh, thank you.
Dr. Carlson, we typically start off the show by giving our guests the floor to talk about themselves a little bit. If you could sort of introduce yourself, take us back a little bit to sort of, where you grew up, how you got interested in science, how you got interested in ichthyology, and ultimately a little bit of your transition into sort of the epicenter of all things regenerative biology, that would really be great.
Well, this could be a rather long story because
that's okay, I have time.
But when I was a kid, I was always interested in animals. In fact, one of my major interests is obviously fish biology. And I think that was kindled when I was 5 years old when I caught my first fish, a sunfish, on a cane pole. But I think things started in the late 1940s when I was living in a suburb of New Haven, Connecticut. And at that point, my father was a Lutheran minister at a church very close to the Yale Divinity School. And we were lucky on Sunday to have a lot of students there or visiting faculty come to our house for social occasions. And I got to meet quite a few very fascinating people, several of whom were missionaries in Africa. And at that point, I wanted to become a missionary to Africa. And I was also very much interested in languages because people came from many parts of the world. And every time someone came, I'd ask them to write a page in their own language, and I kept a notebook of these. So anyway, after a few years, I came to the conclusion that I was much more interested in studying African animals than saving native souls. So I made the decision to go into biology at that point, and really never thought much about it until I went to college. And I went to a small college in Minnesota, Gustavus Adolphus College, and was a majored in biology plus a couple other things. And two experiences there really changed the course of my entire life. And I think it goes to show how seemingly inconsequential things can have a huge effect.
Mm-hmm.
And the first experience happened maybe when I was a junior in college over Christmas vacation. I was living in Minneapolis at the time, and went down to a pawn shop in Skid Row in Minneapolis, just looking around. And I happened to see in that pawn shop a Russian English dictionary for sale for 50 cents. And I said, gee, this sounds like a good deal. I'll buy it. So I bought the dictionary, sat on my shelf for a couple of years, and we can pick that up a little later.
Sure.
So that was number one, that frankly changed my entire life. And then the second experience also happened at Gustavus. For some reason, and I don't remember why, I was on campus the day after graduation, and I didn't have anything to do. So I went into the library, and they happened to have an issue of what was called the Journal of Experimental Zoology. Basically, the article was on the regeneration of the lower jaw in salamanders. And after having read the article, for me, it was very much like a religious conversion. I had already accepted a scholarship to go to Cornell to study fish biology. But I immediately upon reading that article, I said, no, I'm going to go into regeneration. So I went to Cornell the following fall and told my advisor immediately, yeah, I'll get a master's degree in fish biology, but I really want to go into regeneration. So during that year, I spent about 20 hours a week in the library reading about regeneration. So that pretty much got me started into that particular field. But another thing that happened during the time I was at Cornell is that I decided to get my money's worth out of this dictionary, Russian English dictionary that I bought. So I took a course in scientific Russian. And at the end of that course, I was poking around in another used bookstore and ran into a Russian volume on regeneration. Mm-hmm. And I could read a little bit of the language at the time, but I wasn't very good at it. So I could understand the science, but the beginning of every article had language that didn't make any sense to me at all. It was basically political language. So I basically will drop that thread in the story again. And when I was at Cornell, I was advised by a family friend who was the chair of a physiology department that if you want to go into regeneration, basically you should go to medical school, because at the time it gave you a very broad but fairly deep education in the basic biological sciences. But he also suggested to do a double degree. So I applied to the University of Minnesota, and even though it was after the deadline for applications, they accepted me. So that the year after I began my fish studies, I got my master's degree, but then was enrolled in this MD Ph.D. program at the University of Minnesota. Mm-hmm. And my thesis work at the time involved growing extra limbs on salamanders. Now, in a salamander, if you cut a limb off, and often this will happen in nature, mm-hmm. It will grow back again. And the idea that I had, and it wasn't my idea, it was an extension of some other ideas for different purposes, was that, or the question really, and asking questions is really important in science. Sure. Do you actually have to cut off a limb in order to have it grow back? So the experimental model I used was a very strange one. It involved implanting a piece of frog kidney underneath the skin of the limb of a salamander, and that stimulated the growth of a new limb. So that demonstrated that you didn't have to cut the old limb off in order to get a new limb growing. So that was really the beginning of my studies in limb regeneration. And I think we'll just tie up a few knots, because we have to connect Russia with all this. Yep. When I was finishing my studies at the University of Minnesota, I was taking my final oral for my PhD. And usually what happens after you've met with your committee, you go out in the hall and quaver for a while they make a decision as to whether you get your degree or not. What happened with me is I went to the mailroom of our department and opened up a copy of Science magazine. And in the back of the issue of Science, there was a little want ad basically saying that there's a new program, exchange program, between the US and Russian Academy of Sciences. And I got so excited seeing that I went to my lab and started writing a letter to
Mm-hmm.
get information about this. In the meantime, my committee came out to the hall looking for me and couldn't find me. But that was the beginning of a very interesting part of my career, because the day I was supposed to be graduating from the University of Minnesota, which was mid-December, I was on an airplane flying to Moscow, where I spent pretty close to a year studying regeneration and doing research on regeneration in the Soviet Union. So I think I'll stop at this point.
It's somewhat prescient that that you brought that up, because my next question, you know, I've over the years, obviously, I've studied a lot of your work, and sitting here, I do have a copy of Regeneration Research in the Soviet Union from the 1960s, where
Okay.
you, as you mentioned, you spent the time in the exchange program, and it's a fascinating paper because you document, obviously the Cold War was going on. You document that, things were similar between Russian and American regeneration before the war, but then afterwards, the 20 years that followed, things varied a lot between what was happening in America and what was happening in the Soviet Union. And you talk about sort of this greater influence in the microenvironment and understanding not just sort of, I guess, while we were focusing here in the US more on cells or whatever, they were much more focused on sort of the conditions, whether it be in the limb or organ, what have you, that allowed some type of inducible and generation events. Can you take us a little through sort of what you learned on that trip and sort of how that's crafted, what you've done, what you did when you came back?
Yeah, well, I think in order to really get into this, and this again is I'm going to give you a shortened version of a very long story. In that era, science in the Soviet Union was closely intermeshed with politics. Okay. And in order to understand not what they did so much in science, but why they did it, you had to understand the politics. And that got me into all sorts of trouble, even when I was in medical school, because in order to understand their politics, I was subscribing to Pravda, which was delivered to my door every day for $2.40 a year. And that got me in big trouble with American military intelligence and CIA and who knows who else. But anyway, getting back to Russian politics, the post-war Russian politics were totally dominated by the ideas of a fellow named Lysenko, who was a Ukrainian agronomist. And he came up with the idea that, again, to make this very brief, that environment trumped heredity, and that you don't have to worry about genes or even cells for that matter, but especially genetics. And almost every article that was published after the war started out with a denunciation of Mendelism, Morganism, Weissmanism, which was the basis of Western scientific thought at the time.
Sure.
And even odder subdivision of this doctrine, and this is directly relevant to what I did later on in research, was a woman named Olga Lepeshinskaya, who I think was high school educated, but somehow or other she had ingratiated herself with Stalin. And as a result, she wielded enormous influence in Russian biology. And by the way, at that point, Russian biology, or the political influence, was strong enough so that literally wiped out 1 or 2 generations of Russian geneticists. And some of the world leaders in the field, including the leader in plant genetics, literally starved to death in a Russian prison during that era. But anyway, getting back to Lepeshinskaya, she came up with what was called a new cell theory. And the basis of this theory was that you don't have to have cells arising from pre-existing cells, therefore you don't need genetics. And she said, in particular, in the embryo and in regeneration, new cells can arise from what she called a living substance. She actually published several books, which I used to have. Now they're at the University of Michigan Library, showing pictures of new cells coming up from this soup. So we'll drop that at this point. I'll come back to it a little later. But anyway, the bottom line was that much of Russian regeneration research, which had a philosophical backing, going back to Friedrich Engels' dialectics of nature, they basically said that a lot of what happens in regeneration is controlled by your environment. And that fit in perfectly with Lysenko, fit in perfectly with the ideas of Marxism at the time. So at that point, Western biology basically was looking at limb regeneration, at least in this particular field, which was more akin to processes that occur in the embryo. Whereas the Russians suddenly started looking at regeneration in mammals. And this is something that at the time, most Western publications or books said it can't happen. So that there's a tremendous divide in terms of outlook in this particular field, in the philosophical basis for doing research. So when I came to Russia, at that point, I was working in the laboratory of a Professor Polozhayev, who had done some brilliant research in the 1930s and 1940s on limb regeneration in salamanders and trying to stimulate limb regeneration in frogs. But during the early 1950s, he got into some big trouble, and some of my Russian colleagues told me that he was an idealist when he should have been a realist. And as a result, he was put in a mental institution for a couple of years. And when he came out, he frankly wasn't the same, but he still did some fascinating experiments involving mammals. And during the early 1950s, he came up with a technique for regenerating big holes in the skull, which normally won't regenerate. And this actually preceded some work that was done in the mid-1960s. It was considered breakthrough in the Western medicine, but he had actually discovered this a long time before, but because he had to publish in Russian, nobody knew about it. So anyway, I found myself in Polozhayev's laboratory trying to do experiments on seeing if I could grow extra limbs on axolotls, which is a big salamander. They're normally found in the water near Mexico City. Whereas earlier I had worked on newts, which are cute little things that I would get from New England. And during that time, my research, frankly, didn't amount to too much, but the most important part of the whole period when I was there is that I became acquainted with pretty much everybody in the Russian field of regeneration. And for many years, I ended up being basically the funnel between Russian or Soviet regeneration research and Western research, because I knew the language, and I was able to get access to material from the Russian literature that nobody else had a chance to see. So it worked out pretty well for quite a bit of time. I think I'll stop at this point with historical background again.
No, it's completely fascinating. And I mean, it's just thinking about sort of the what you had to go through during the middle of the Cold War, actually, and to sort of get this knowledge, or sort of merge these two knowledge bases is completely fascinating. You know, when one looks at limb regeneration, so we look at this, and this is obviously something that, as you're mentioning, you can go back in the literature and you see resources from when, I mean, I was been studying limb regeneration in one form or another for technically a couple hundred years, but really a lot was going on, which we'll get into a little later on, sort of in the early 19th century, the early 20th century, in regard to, it was well before we knew a lot about genes, but sort of one of the biophysical aspects of how things happen, and we'll get into morphogenesis in another question. But looking at things today, which we are in this very what I'll call stem cell centric era, where in many ways a lot of regeneration or regenerative medicine is reduced to sort of cells now, like, once again. I mean, how are you viewing sort of the state of the art now in 2020? Do we feel that so we've left a lot, and you obviously in the book highlight whether it's biomechanical forces or electrical fields, or many of the things that you touch on in the book. Do you think that in 2020 sort of the regenerative medicine space is doing enough on these various fronts, or have we sort of reduced it a little too much once again to this sort of the stem cell centric model, in your opinion?
Well, I would agree 100% that we've reduced it too much. Not that stem cells aren't important, but again, just for a little historical perspective,
Please. Back
when I got in the field in the early 1960s, if anyone talked about stem cells, they would be laughed out of a meeting. Nobody believed in them. And I still remember a few papers, when, especially people talking about blood-borne stem cells, that was considered absolutely crazy. So things come in and out of fashion in science. And right now, stem cells are very much in fashion. But I think with respect to what you were saying, one of the positive notes about the whole stem cell field right now is the connection between stem cell biologists and bioengineers, because the bioengineers are the environment component and the stem cell guys are the genetic component. And without having a combination of the two of these, one isn't going to make much progress. And particularly now, and this is not only true in regeneration, but in other areas as well, mechanical influences on gene expression are becoming extremely important. And we're just beginning to understand what that's all about.
Absolutely. Yeah. Agree 100%. I'm glad you agree with me. I feel like I'm validated in that sense. So moving then into an area, there's a fascinating quote in one of the chapters in the book where you get into morphogenesis, and it basically goes like this: Carlson, stay away from morphogenesis. It's a bag of worms. And this was from 1966 by Dr. Charles Steed Thornton, himself another regenerative biology luminary in the area of denervation and epimorphous regeneration and so forth, telling you, 50 plus years ago, this is a tough area, but it's 2020 now. And once again, a lot of what you point out in the book in terms of what I'll refer to as sort of regenerative laws, the things in nature that, in terms of proximal distal regeneration dynamics, how wounds heal by going the shortest distance as opposed to the longest. A lot of these things that, people documented in 100 years ago, don't hear much about them for some reason, but they're sort of underlying the sort of dynamics. You talk about morphogenetic fields that people talked about in the 1920s in the book. A lot of this stuff seems extremely relevant today as we're, once again, playing with this sort of stem cell model. Talk a little bit about the history of morphogenesis as you were getting involved in it, and sort of where you think we've come and where we need to be going in terms of this really crucial concept of form, in the sense that, there's no gene in here that says, a hand looks like this and should be shaped like this. There's really complex systems all coming together. Talk a little bit about this really, I think, unexplored area.
This is still a really tough one.
Yeah.
And the bottom line is we still really don't understand the basis for morphogenesis. We know a lot more than we did 50 years ago, but we don't really know if there's such a thing as a genetic code underlying the sort of morphogenesis that occurs when you're an embryo, or what happens when a limb is growing back. But for a long time, I was sort of straddling the fence between regeneration in amphibians, which we call epimorphic regeneration, and regeneration in mammals, which we call tissue regeneration.
Mm-hmm.
And it turns out that the two regenerative processes, even though at the cellular level, sometimes they're very close together, morphogenesis operates under quite different laws. And in mammals, morphogenesis is highly directed by physical factors,
mm-hmm.
whereas in the amphibian regenerating limb, it's directed by other factors, which we frankly still don't know an awful lot about. In fact, there's an interesting, and again, so as a result, a lot of people in the field came up with models trying to outline the parameters of morphogenesis, but in terms of understanding exactly what happens, in some areas, like, for instance, the development of the hand in an embryo, we now have a pretty good molecular explanation for almost everything that's going on that we have asked about. But there's still some fundamental questions that we don't know, particularly in the area of regeneration. And just as an example of how people got into the field back 50, 100 years ago, it was mostly through accidents. An accident would happen, something odd would take place, and then you try to figure out why it happened. And I'll give you a personal example,
Sure,
just to show you how people used to get into the field of morphogenesis. Back, actually, when I was a graduate student at Minnesota, I was very much interested in, at that point, molecular biology, and I still remember taking a course in nucleic acids when the genetic code was in the process of being discovered. And every week my professor would come in and say, we now know the code for another amino acid. But one of the big molecular tools at that time was a drug called actinomycin D, which was considered to inhibit DNA-dependent RNA synthesis as if there wasn't anything else. But so anyway, I was able to get a hold of some of this drug from George Hitchings, who was one of the developers of the drug, and injected it into salamanders to see what would happen. Well, I injected it into salamanders, into newts, and certainly regeneration was inhibited, but the animals were so sick that it was hard to really differentiate between overall pathology and the effects of the drug. So later on, after I was at the University of Michigan, I decided to do a little more refined experiment. And at that point, I was working with these axolotls, which at that point were about 6 or 8 inches long. Mm-hmm. And instead, so in trying to eliminate the systemic toxic effects of actinomycin D, I decided to do something a little different. And that was to take the skin off of the limbs and soak the skin in actinomycin D, and then put the skin back on the limbs, and then amputate the limbs and see if the regeneration process was inhibited by the actinomycin D. Well, it turns out that really didn't happen, but I ended up getting a couple regenerates that were basically upside down. So instead of the palm being on the bottom, the back of the hand was on the bottom and the palm was on top. And I asked the question, well, what was going on? Did I make a mistake in terms of putting the skin back on? Did I put the skin back on upside down by mistake? So I did an experiment where I purposely took the skin off the limbs of axolotls and turned it 180 degrees, and then sewed it back on, let it heal, and then amputated the limb. And to my surprise, the limbs did regenerate, but they regenerated with up to 15 or 17 digits and extra hands. In the normal hand has only 4 digits. Mm-hmm. So obviously at that point, something very strange had happened. And that started me on about a close to 10-year period when I was investigating a particular morphogenetic model, trying to understand why just simply physically turning the skin around would cause extra limbs to form when the limb regenerated. So what I did was more what you would call reductionist research, trying to see what there was in the limb that contained what I called morphogenetic information. And I tried rotating every tissue in the limb that I could, and found that if you rotate the dermis of the skin or the muscles 180 degrees, it would cause the formation of an extra limb or a whole bunch of extra limbs and digits, whereas epidermis, bone would not cause any change whatsoever. And at the time I was doing this, several other people were working on different models where they were also getting extra limbs forming, but from different mechanisms or using different surgical procedures. And that started a period of morphogenetic modeling, which was at its peak in the mid-1970s. And I didn't get into this aspect too much. I was more interested in what there was in the limb that would do this. But some very sophisticated models were developed, and they explained an awful lot of the phenomena at a superficial level, but they did not explain it at a molecular level. Mm-hmm. And we're still, at this point, trying to understand at a molecular level the basis for some of these morphogenetic phenomena. We now know, at that point when we were doing this, we really didn't understand what growth factors were at all. And now we know that a number of very powerful growth factors, one of which is called sonic hedgehog, is very important in terms of forming extra limbs. But we were really flying blind at that point. And that's why gross modeling rather than molecular approaches worked. We're now just at the point in the last 10 years where people working on salamander limb regeneration have the molecular tools to get at this type of thing.
Fascinating, fascinating. Leveraging off of that for a moment, there are a variety of fascinating papers from, I forget what decades these were from, and you mentioned some of them in the book. They are related to obviously there's this double-edged sword that's always existed, and we hear about it nowadays all the time, between sort of regeneration and oncogenesis, and sort of some type of balance existing between the two if you tilt that seesaw in one direction or another. Yet there are these fascinating papers from the 30s, the 1940s, that show this, what are referred to as sort of tumor reversion in the regenerative microenvironment. We see these papers in frogs and limb regeneration. Later on in the 1970s, the first examples of this here actually in Philadelphia at Beatrice Mintz's lab at Fox Chase, where she showed sort of tumor reversion in developing mammalian embryos. Could you talk a little bit about sort of this regeneration cancer axis, let's say, and some of these, once again, because even in the papers nowadays that reference these papers, they talk about these morphogenetic normalization or embryonic fields that normalize tissues. Can you talk a little bit about some of the learnings on this particular front? Because I think they're forgotten way too much, and they're fascinating learnings in regenerative biology.
Yeah, I can't talk too much about them. I never worked in the field, and my basic sense on this is that at one level, particularly when you have a very active developmental process, it can affect, at least in animals that are capable of epimorphic regeneration, it can influence the course of a tumor. I don't know in the case of, for instance, like a sarcoma, in a mammal, whether a highly regenerative environment would have any influence whatsoever on that. Beatrice Mintz did some fascinating experiments, if I remember right. I don't remember the specific details of a lot of her things, but the other thing that it was almost the reverse. Tumors could influence regenerative processes. And again, now in retrospect, it's probably because they contain growth factors.
Right.
And I think, again, what could easily have happened in some of these tumor experiments is that in a highly charged environment full of growth factors, the growth factors in the developing systems might be able to suppress some of the things going on in tumors. That would be, I think, probably a contemporary interpretation.
Okay, I appreciate it. I was just, as I said, putting it out there because there is a sort of this fascinating intersection, and we forget about it sometimes, but those papers exist, and then they're just really interesting dynamics that were not fully described in these papers, of course, but the sort of the phenomenological,
But some of these old papers will be rediscovered, and again, with more contemporary tools, I think some fascinating explanations will come up.
Sure. Along those lines, though, and once again, you have a wonderful chapter in the book on aging, and this is a topic that we discuss on a lot of our shows, aging and sort of the this longevity biotechnology movement that's occurring nowadays. And in the book, you talk a little bit about what I'll refer to as the cross-age transplantation era, where there was a lot of once again, I don't know, the 50s, 60s, whenever, but we're moving things back and forth, taking, an old heart and putting it in a young body, and a young heart putting it in an old body, so forth and so on. And once again, you talk a lot about that, once again, it's not always the youthfulness of the thing you're putting in, but how the microenvironment of the system affected whether that young heart stayed young or not. Could you talk a little bit about some of the learnings in regards to aging as it pertains to regenerative biology and some of this history?
It's interesting that at present, if we look 2 feet away from me, my desk is covered with papers. I'm writing a book on muscle biology right now, and I'm writing the chapter on aging.
Excellent.
Muscle. But I think before we get into the aging of muscle, which is a fascinating story, I probably should go back again many years to the whole field of muscle regeneration.
Please.
Because back when I got into the field of regeneration, every textbook in basically in the English language of surgery or histology or pathology said the skeletal muscle cannot regenerate. even though muscle regeneration had been described in the German literatures, I think in 1887 was the first paper, if I remember correctly. So it was much to my surprise when I was working in Russia that I visited the laboratory of a fascinating fellow named Alexander Nikolaevich Studitsky, who had come up with a fascinating model of muscle regeneration, which goes back to politics and back to something I talked about a few minutes ago. Remember I mentioned Olga Lepeshinskaya and her new cell theory?
Absolutely.
Well, Studitsky, who is a very political animal, but a brilliant classical biologist as well. This is back in the late 1940s when officially this Lysenko doctrine took precedence over real science. He decided to test this new cell theory of Lepeshinskaya by taking what was called a post-mitotic tissue, skeletal muscle, and said, if I can totally destroy a skeletal muscle and get it to grow back again, it will prove the validity of Lepeshinskaya's theory. So what he did was to take the biceps muscles of chickens or the gastrocnemius muscle of rats, chop them into little pieces. In Russia, he says kasha, which is like Russian oatmeal, into pieces small enough so that there wouldn't be any surviving cells or nuclei. And he said, okay, if I do this and if I get a new muscle, it's going to prove that Lepeshinskaya's theory is correct. So he did this, and surprisingly enough, he came up with new muscles within the limb of the rat or within the wing of the chicken, and spent about 10 or 15 years investigating this phenomenon, but always interpreted his results as showing that this new muscle came from this living substance, non-cellular living substance. And yet some of the experiments he did were fascinating. Well, when I was in Russia, I visited his lab several times and realized that he had discovered a remarkable phenomenon. So when I came back to Michigan, I spent about a year and a half trying to repeat his experiments, first on frogs and then on rats. And turns out it worked. But I basically put the results in the context of Western biology rather than the context of Soviet biology. So I wrote a monograph on that I think was published around 1972. And at the same time, in the early 1960s, a biophysicist at the Rockefeller Institute, Alexander Morrow, discovered a very unusual cell with the electron microscope that was located between a muscle fiber and the little membrane that surrounds the muscle fiber. He called it a satellite cell. And he suggested that possibly this might represent the cellular origin of healing or regenerating muscle. So these two things came together. One, a possibility of a stem cell for regenerating muscle. And then the other one was the fact that you can regenerate an entire muscle badly, but at least it regenerates, which came from Russia. And for about 15 or 20 years, maybe more than that, I'd say maybe 20 years, there was a huge argument going on among the people who worked on muscle regeneration as to whether the new muscle came from stem cells or satellite cells, or whether it came from what's called de-differentiation, the breakup of an existing muscle fiber into smaller units. Well, it turns out after a lot of arguments, and I was at some fascinating meetings where people were literally screaming at one another, it turns out that both are partially correct. And I find in so many scientific arguments, if you have good scientists on both sides, each of them has some element of truth.
Sure.
So it turns out that in mammals, the source of regenerating muscle is satellite cells, or these stem cells.
Okay.
In the amphibians, in the amphibian limb, it gets extremely complicated. In some species, de-differentiation or the breakup of muscle fibers is the mechanism. In other species, the amphibian equivalent of satellite cells is also takes part in the process. So as it turns out, both sides were partially right within the context of the systems that they were studying. So muscle regeneration continued, and it was of great interest to the people, especially in the Muscular Dystrophy Association, because it turns out that the basic problem in muscular dystrophy is a genetic problem where a huge protein called dystrophin is abnormally put together, and it makes the muscle cell walls weak. So when kids who have muscular dystrophy move and stress their muscles, the muscle cells break. And these broken muscle cells then regenerate, and they'll basically initially regenerate quite well, but after a long period of time with repeated breaks in the muscle fibers, the muscle cells essentially run out of gas or run out of stem cells, and the kids end up having their muscles infiltrated with fat, and then they often would used to die in the late teens. But that gave an impetus to a lot of people to study muscle regeneration. And at the same time, and this is more personal with me, some plastic surgeons came up with the idea of transplanting entire muscles as free grafts to try to deal with a condition called facial paralysis, where this part of the face, if the facial nerve was cut, all the muscles would be flaccid, and the mouth would go down like this, the eyes would be open, and you had no facial expression on this side. And at the time, again, this is a long story I won't get into, but I was working in Czechoslovakia, in Prague, with Professor Ernest Gutmann, who is sort of my scientific godfather. And we were interested in muscle regeneration and trying to come up with models to study the physiology of regenerating muscle, and started out using this minced muscle technique of chopping up the muscle. But it turns out that a better technique was to take a small muscle in a rat and graft it as a free graft. So you take the muscle out of the limb, cut all the connections, put it back in again. You sew the tendons, but let the blood vessels and the nerves, they have to grow back into the muscle. Well, when you do that, the muscle breaks down and then grows back again. So that makes a good regeneration model, which I had used then for about 30 years as an experimental model. So these all came together for me. And this gets back into the aging, because back in the 1980s or late 1980s, I was working with a physiologist at Michigan, John Faulkner, who was very much interested in aging muscle and muscle contraction. So we formed a collaboration where he did the physiology, I did the anatomy, morphology, and did the surgery. And using muscle transplantation as a technique, we were interested in why old muscle regenerates so much more poorly than young muscle. And fortunately, I had a colony of rats that at that point was about 70 generation inbred, so we could move tissues from one animal to the other without rejection. So we decided to try a model of taking very old rats, 24-month-old rats, which would be maybe the equivalent of a 70, 75-year-old human, and put them into young rats. And then take a muscle from a young rat and put it into an old rat, and basically compare results and see what would happen. Well, to make a long story fairly short, if you put an old muscle into a young animal and let it regenerate, it regenerated as well as a young muscle regenerating in a young animal. If you put a young muscle in an old animal, it regenerated just as badly as an old muscle in an old animal. So the bottom line there is that the environment in which the muscle graft was regenerating played a dominant role in the success of the graft. And we did a batch of other experiments, and one of the major contributors to this phenomenon was innervation, because that's part of the environment. And it turns out that reinnervation of a regenerating muscle is much more successful in a young animal than an old animal. And at that point, we were pretty much thinking that the environment was 90% of the solution, if not even more. Well, now we know, and now I'm finding out, since I'm writing this chapter on aging muscle, that there are also aging changes in the satellite cells or the stem cells of muscles themselves, so that this is an intrinsic aging issue. So we have both an environmental stimulus or influence plus an intrinsic influence, and both of them work in terms of aging to cause a poor result if an aging tissue like muscle or something else is going to have to heal.
Fascinating. It's so fundamental and important in sort of everything that's going on nowadays in this particular field. It's one of those areas that anytime anyone talks about aging and sort of, all we need to do is put young stuff in. Sometimes it misses the big. I always say you got to go back and you got to read what Bruce documented years ago.
Yeah, I hate to say it, but the main message is it's better to have a young body than an old body.
Exactly. It's a good point. Moving on to something a little more esoteric, but I would love to explore this with you. You are working with the Fetzer Franklin Fund, which it says on the website that focuses on foundational questions of the frontiers of physics, biology, and consciousness research. I think about while you were spending your time at the University of Michigan, next door in Indiana in the late 60s was Dr. Paul Piche, another anatomist doing some really interesting work in terms of shuffling brains of amphibians and all sorts of interesting things on the central nervous system front. And finding, it was pretty difficult to destroy memories, even if you can mash the brain up a lot. There's been some work on this front in more recent years in terms of, how do we smash this brain apart, and why does this little salamander remember so much stuff? I'm not sure what you're doing at the Fetzer group, but are these the areas you're exploring, or can you talk a little bit about this area if it's something that's interesting to you? I'm not exactly sure of how you're connecting biology and consciousness currently, but would love to hear your thoughts on this really interesting theme.
Well, this is another weird story that
I like weird stories.
Been connected with the Fetzer Institute for about 20 years, and the really two entities, which I won't get into, but basically the Fetzer Institute was started by John Fetzer, who was a radio television pioneer who also ended up owning the Detroit Tigers for a long time. Mm-hmm. And he was a very complex individual, but had a very interesting personal life. He was very much interested in spirituality, and it was the sort of thing in love. And as a businessman in the 1950s and 60s, you didn't talk about that sort of thing. Yeah. But he had no children. But when he died, he put his entire fortune into this Fetzer Institute, which was basically at the time looking at esoteric science and also spirituality. And the reason I got involved with them was that back, I think it was 1989, I was on the medical school executive committee at Michigan. And after one of our meetings, the dean came up to me and said, I have a friend who's a president of this outfit called the Fetzer Foundation at the time. And he's having a meeting with a bunch of Russian scientists and needs to find someone who can talk to Russian scientists. And he said, would you mind going over there? So I went to Kalamazoo, Michigan, had the meeting with Russian scientists and talked with a few people there and got connected with them, and I've been with them ever since in some very different capacities. During the 1990s, I was involved with what they called the Bioelectrodynamics Committee or Bioelectromagnetics Committee, where they were much interested in a phenomena like this because of John Fetzer's interest. And that was a period, sort of at the end of the period, where people working on limb regeneration or bone regeneration were very much interested in bioelectrical effects. Mm-hmm. Things progressed from there to what we called biodynamics, where we were interested in phenomena such as emergence. Mm-hmm. But then around 2000, they asked me to be on the board of the Fetzer Institute, which I've been ever since, and also the Fetzer Memorial Trust, which is the entity that contains the Fetzer Franklin Fund. And this fund has really had a very interesting history. They are trying to get people in various scientific fields to look at different aspects from the current model, so to speak. Mm-hmm. And one of the areas that has really been heavily influenced by Fetzer Franklin is quantum physics, where David Bohm, who is one of the pioneers in a lot of elements of quantum physics, actually, interestingly enough, spent a year at the Fetzer Institute, which I hadn't realized for a while. But anyway, one of the main interests, long-term interests in this whole area is consciousness, which, depending upon who you talk to, people would say it either does not exist or it's one of the great unanswered questions in biology, or in science, really, not just biology. Sure. So right now we're really looking at how one might, as an institute or a foundation with a relatively limited budget, how we might potentially have an impact on this field. And we haven't even decided the extent to which we're going to really be heavily involved in the future, but in a couple of years we'll make that decision.
Excellent. I'm glad somebody will be looking at it because it is a tough one.
Well, yes, and it's the kind of thing right now that governments don't fund. And
Right.
if you're a foundation, what you want to do is try to influence the field at the point, to allow it to develop. And I always use the analogy of being in the crest of the wave, but you don't want to fall off.
Sure.
And once you get to the point where a field is well enough established, then governmental agencies will fund it. And then the foundation gets out of the way and gets into something else. But that's the way foundations typically work.
Sure. One last question I have. You've aside from everything you've done in the area of regeneration, regenerative biology, and so forth, you have also had this passion for lake biology. And you've written a book. Are there learnings? Once again, we as we talk about sort of the microenvironment in regeneration and sort of these hierarchies that exist in the human body, from genes to tissues to organs and so forth, are there interesting learnings from your lake biology research in terms of studying an ecosystem in this context that are useful in the sense of whether it's regeneration, global warming, whatever it is, that you might want to talk about, because it's an area I didn't know you published until I went on Amazon the other day and saw this book. But tell us a little bit about this as well.
Yeah, well, this goes back to my passion for both fish and fishing, and actually every year. As soon as I started working, I would always take a month off and go to our family cabin in a lake in northern Minnesota and go fishing. And I won't get into fishing techniques at this point, but by the mid-1980s, I'd come up with a few very unusual techniques for a particular fish called a walleye, which at that point, most people would fish for at the bottom of a lake, which they still do. But just by accident, I found out that I could catch them on the surface in 200 feet of water, and wrote an article about this and sent it to a major fishing magazine. And they didn't believe me until the editor came out, and I took him out in a boat, and we were able to catch some fish that way. But that started about a 25-year career of writing for a fishing magazine. But since I retired, I've gotten back into this. I closed my laboratory down when I retired, and then I'm back to my second scientific childhood, which is fish biology. And again, a chance event managed to influence an awful lot of what I've done since retirement. And this would go back, I think, about 12, 13 years ago, when I just published this book on lake biology. And then there was a group of state senators taking a tour of northern Minnesota, trying to see what was being done with some funds they had to restore the environment. And I was asked to give a talk about lake biology to this group of people at the public landing of our lake. So I gave the talk, and everybody left except one fellow who said, "I have something you might be interested in." And it turns out he was name is Dallas Hudson. He was a technician for the U.S. Geological Survey, which has a project on a lake near our cabin, a 30-year project where they're trying to look at every environmental influence that can affect a watershed.
Mm-hmm.
And he had been doing a surreptitious tagging study on northern pike for about 5 years, trying to look at things that influence the growth of these fish. And he says, "Would you like to see what I have?" So I went to his office at their field station, and he showed me his database, and I realized it was just a remarkable database on fish growth. So at that point, I said, "Yeah, this sounds like something we should really pursue," because it was a lake with no cabins, no public access, so it's like a laboratory. So we contacted his supervisor and said, "Should we really put together a formal project on this?" And we all agreed it would be a good idea. So because I have a background in this area, was the official director of the project, which we got approval from the Minnesota DNR. But for 10 years after that, we were doing a study on the growth of northern pike, and we would catch pike, tag them, measure them, take scale samples, and just literally follow the growth of individual fish, which has rarely been done because you normally won't have the opportunity. And to do this, what you do is, when the ice is just leaving the lake, you go out and set trap nets in this ice cold water and catch pike as they're spawning, and you tag them, and then you get numbers, and then in order to do proper statistics, you have to collect them again later in the year. Using some proportionality, you can determine the population of the fish. Well, it turns out that the best way to capture pike and release them later in the year is sport fishing. So I had to have the sad duty of 2 days a week going to this lake for 8 hours a day, sport fishing for northern pike, trying to catch as many as 40 pike a day, and then measuring them and tagging them and so forth. So we did this for almost 10 years. And part of the project involved a northern pike specialist in the Minnesota DNR who was interested in behavior. And we were implanting devices in the bellies of big and small pike that every half hour would record the depth that they were at and the temperature they were at. And I still remember for 3 weeks, it's the only time I've ever done surgery outside in 8 inches of snow. And by the end of that, I couldn't feel my fingers for the next month. But anyway, we amassed this enormous database of pike growth, and we're just about to finish our project when the Department of Interior, which runs the USGS, closed down many of the projects involving climate change for obvious reasons in this political environment. So that put a stop to this particular project. But the thing you learn when you're doing ecological research is that everything affects everything else. So the environment is critical. Yep. And right now, as a matter of fact, I'm just starting a brand new project. We discovered last fall that there are zebra mussels in our lake. This is a nasty invader that completely changed the ecology of a body of water. And these came from Central Europe and have progressed through the United States. So it's a real pest in about a third of the states in the United States. And our particular lake is one that's been heavily studied for some unique reasons I won't get into here. But we have a huge database already, and what I'm doing is working with the folks in the DNR to try to put together a study where we're going to look at the effects of these zebra mussels on the overall ecology of a lake. And one of the things I've been doing, and I started last summer before I even knew we had zebra mussels, was using underwater cameras and looking at huge stretches of the bottom of the lake. And now we have a before picture, and now we're going to follow this over a number of years to see how zebra mussels really affect populations of anything from plankton to fish. Mm-hmm. Mm-hmm. But here again, the environment plays such an effect on things. It's not necessarily the genetic basis, although it might, but it can affect all sorts of things.
Sure. I appreciate that. I mean, it's once again, I'm sitting here fascinated by just the amount of stuff that you've accomplished, and once again, I think the word nowadays is sort of this convergence, and the fact that you're taking all of these different disciplines and putting them together in this context, whether it's regeneration, lake biology, consciousness. It's really an amazing suite of things that you're doing, and just a great story, and really, thanking you for doing it, because I think it's so fundamental to where we're going forward from here. For everybody that is going to be watching this episode and listening, we've been speaking to the amazing Dr. Bruce Carlson, Professor Emeritus, Anatomy and Cell Biology, University of Michigan, scientific advisor to the Fetzer Franklin Fund. If there's one resource I could encourage you all to pick up at some point, if you want to learn about the space, you want to learn about regeneration, regenerative biology, aging, the history of all of the above, and and you only have money for one book, I suggest you pick up all his books, but I call it the Bible. It's really an amazing story that you have, Bruce, and everything you've done. We say on the show, thank you for moving the human story forward in this way, and thank you for everything you've done and continue to do. But this was truly an amazing honor to have you, and just thank you for taking the time, especially in today's crazy environment we find this in, for taking the time to just come on the show and share your knowledge with the world. It's really great. It's really been an honor.
Yeah, thank you very much, Ira.